In this lab, we will talk about three main types of histology for the accessory digestive system. The first of which is going to be our paired salivary glands. Now, these tend to be the trickiest structures for students to absorb. The reason for that is we have three types of salivary glands and they all look different under a microscope. And yes, you are expected to know those differences. So before we jump into the three types, I want to talk about the basic structure of a salivary gland. So over and over again today, we're going to see this berry-shaped pattern, and what we're dealing with is berry-shaped clusters of cells, and these are called acini, or acinus for singular. We are going to have two types of clusters. We're going to have mucus acini and serous acini. Mucus acini are going to secrete mucin, and if you think about the name, mucus, it's slimy. Mucus and mucin are very similar. So what these mucus acini are doing is secreting this lubricating mucin. And that's going to help lubricate things in our mouth and protect us from pathogens. Whereas serous acini are going to have a more serous or watery function. So they're going to secrete proteins and this really watery fluid that helps to break down starches in the mouth. Other things we will see over and over again are intercalated ducts. So anytime we have secretions from a gland, we're going to probably be dealing with a duct. And we can see some of those things on this slide here. So there's several different lumen within this just generic salivary gland. And so what we're dealing with is lots and lots of these ducts that are going to collect the secretions. So for example, with the parotid gland, we have a parotid duct, which is going to send that saliva to the oral cavity. We will also be dealing with mixed glands. So sometimes you'll see glands that have both serous and mucus cells. And we're going to look for that pattern today on some of our salivary glands. First up, we have our submandibular gland. And in this case, we are going to use the Yale Histology site. I think this site does a really good job of breaking down the types of histology you can expect to see. You can also practice with these by hiding the labels. So I would recommend that. Practice that recall by figuring out what type of slide this is, and then you can check your work. The submandibular gland is going to be a mixed gland, and that's why I like to show this one first, because it gives examples of both types of cells. We're going to have darker stained serous cells, which are in this dark purple here. I'm going to take away our labels so we can see a little better. So dark stained serous cells, those are producing those watery secretions that help to break down starches, and they are going to be surrounding these lighter mucus cells. So the mucus acini, those berry-shaped clusters, they're going to be surrounded by kind of these half moons of serous cells. And that is what creates this pattern of dark serous cells and then lighter stained mucus cells. We have a sublingual gland. This one is different because it's mostly mucus secretions. So right away you can see this difference in stain color where we have lots and lots of these light stained mucus cells. And that tells us that the sublingual gland is going to have mostly that lubricating function of producing mucin that is going to lubricate the oral cavity. Here we have another example of a duct. So we'll see this pattern over and over again where they typically have a lot of cuboidal or columnar epithelium surrounding a small lumen. Last up, we have our parotid gland. And again, we have another color change. So let me switch back to sublingual real quick. Note we have these lighter stained mucus cells. And now with the parotid gland, we're switching over to a mostly serous gland. We have tons and tons of dark purple tissue here. Again, we have a set of ducts. So we've got 1, 2, 3 ducts all in a row. And then most of this tissue, which is dark purple, is going to be those serous cells. Lastly, you might also see this term myoepithelial cell pop up. If you note here, we've got a little cap of darker tissue around the cell. Now, for this, I want to refer back to our PowerPoint. And I actually really like this slide. It explains well what's happening here. So with some of the glands we talk about, they have this kind of quasi-muscle cell. And you'll see this term myoepithelial. So we've got, in this case, mucus acini, those berry-shaped clusters, and then we have this little cap of myoepithelial cells. So based on that term myo, this is going to have a muscular or a contractile function. So the myoepithelial cells that cap off the berry-shaped clusters here, they're going to contract and help squeeze secretions from these cells into ducts. So they have that additional contractile function to help secrete as much as possible from these cells. And so that is all that term refers to on the Yale Histology site. You will not be tested on that, so let's go ahead and summarize what you need to know for the lab exam. So we talked about the basic structure here, these berry-shaped clusters of acini that you'll find on any of these salivary glands. You need to be able to distinguish between the three types. So parotid had mostly serous cells, indicated by the dark purple tissue here. Sublingual had mostly mucus cells, which are lighter stain, and then submandibular was that guy with both. And we've got a really nice pattern here from the Yale Histology site. Your models have limited images here. So we have just one example of the parotid gland. This is not purple, as you'll note, so be careful with your colors. You want to understand that these serous cells are going to be darker stained, but they won't always be purple. And so we see an example of that here. In this case, the darker red we see is going to be those serous clusters, those serous acini. So when you are studying for the lab exam, I would definitely recommend using the Yale Histology site for the salivary glands, and then the two other types of tissue we will look at in the next few videos have really good resources on the Human Protein Atlas.